I've got a question I'll ask,
Okay,
but it's more about the paper. It's not like logistically about the quiz.
that's fine. We can get into the paper. I don't think
Just wanted
anyone
to,
has
yeah.
any quiz questions, so go for it.
Okay. I was having a hard time understanding why the no litter group had more endophyte diversity.
Mm.
That didn't really make a lot of sense to me. I can't really figure out why the no litter would have more diversity other than maybe like competition. Um, but yeah, I'm not quite sure.
Yeah, I think that freaked out the authors as well, from what I can tell, reading this and then an interview with the lead author, Natalie Christian. So it doesn't really make sense logically from the outset, but you step back and think they took sterile seeds and then planted them in the soil. As they germinate, they start growing up. The first thing they encounter really early in life is that if it's the leaf litter, then maybe what's happening is that they're picking up some microbes really early on that kind of just begin to dominate the microbiome of the plant. If they don't pick those up as they're kind of pushing through the leaf litter, maybe what's going on is that they're kind of more of a blank slate for all that random environmental kind of noise fungi that's coming out in the rain, blowing in on the wind, getting carried there by bugs, that kind of thing. So maybe.
Yeah, that's what I was thinking when I said competition.
Mm-hmm.
So is it just kind of, if there's no litter, then there's more of a chance that like more species can all kind of latch on to the host all at once, and they're not like dominating the space. They kind of like
and it's
together.
Probably a more random assemblage of organisms that are just blowing around in the wind or falling through the rain versus a more defined assemblage of things that are already in the litter. So there'd be more likely to be fungi that are good at being endophytes in that plant or any plant, for example. I don't know. It kind of freaks me out a little bit also. But I think what it's getting at is the idea that the things that determine the assemblage of microbes in a microbiome for any organism are still pretty not well understood. So there are a lot of random processes going on, especially with things that get their microbiome from the environment, like people or dicot plants. So maybe that is kind of what we're seeing happen here.
Yeah, okay, thank you.
I would say we're still in our infancy of understanding how microbial communities form on a host.
Hey, Dr. Chandler. Sorry, Dr. Dr. Zahn. Sorry.
Yeah.
Chandler posted something saying that she wasn't going to be in class today.
Okay.
She had some questions.
I missed that.
It's all right. It's near the bottom. I can read those questions if you want me to.
Yeah, go for it.
She said, this study was published in 2017. Has the agricultural industry put this information into work, or is more testing necessary? That's her first question.
Well, to get it, that's a big question. The answer is they're trying really hard to get this sort of thing into play. So there are several companies. The biggest one that I know of and have interacted with a little bit is Indigo Ag. And they're very much running with this idea of giving plants a healthy microbiome. And they're hiring hundreds of people to work on this question of how do we set things up so a microbiome forms the way we want it to. We're not super far on that, but the feeling I get, and from talking with other people, because this is actually kind of my area of expertise, I suppose, in mycology, is microbiomes, especially with plants. So I'm kind of getting the feeling that we're on the cusp of making a few breakthroughs, not solving all the problems, but we're starting to develop decent hypotheses for a general theory of how plant microbiomes assemble. So it's still left to test those and see how applied they are to diverse systems and all that. Right now, the ag companies that are pouring tons of money into this are really just focused on a few crops. And they're doing that for a few reasons. One is only a few crops are super cash crops that make a ton of money. And if we limit the diversity of plants that we start testing, we're more likely to find things that work on a certain plant. And that's really what they're interested in. From a more academic perspective, we're much further away from finding generalized rules for plants in general and fungal communities assembling.
Okay. So her next question is, the paper mentioned other papers that altered the Jansen-Connell hypothesis in other species as well. I also wanted to add that while reading this paper, I was thinking about the maternal microbiota being passed to a newborn, and then it mentioned it at the end of the discussion. If we have fungi living in us, that usually plays nice. Are the fungi in us considered endophytes? I kind of have the same question. So if we have something, if we get something, and as far, sorry, if a mother gets something and can they pass it on to the infant inside their, if they are pregnant, and essentially have that part, would that be considered an endophyte, I guess, is what she's asking.
Well, endophyte means inside a plant, so no, not endophyte.
Okay.
But we do have commensal or even maybe beneficial fungi living on and inside of us. Now, keep in mind that as far as we know, there's no vertical transmission of microbiomes in humans. So
right. I remember we talked about this last time.
Yeah, it's infant humans are born, their gut is sterile, essentially. There's no microbes in it. And so by the time you're 1 year old, you're packed with E. coli and all kinds of bacteria and stuff in your gut. And that's just from things that you've picked up from the environment. So a while back, it's, I don't know, 10, 15 years now, somebody was looking at this question in humans and basically asking, where is it specifically that humans get their gut microbiome? Like, what part of the environment are they getting it from? Is it from breathing, from eating food, all that stuff? And this guy experimented on his own children, which is, I guess, acceptable. But he had a brand newborn and started taking stool samples and measuring the bacteria types around his house all over the place. And where do you think the E. coli came from that wound up in his kid's gut? He did some pretty nice deep sequencing, so he was able to get specific bacterial strains and basically track exactly where it came from.
I don't know, maybe the baby picked up something off the floor or something, put it in his mouth?
Yeah, it was from the dog. The family dog version of E. coli is what wound up in his kid's gut.
He wasn't one of those guys that said, don't bite the dog. He just said, go ahead and bite it and see what happens.
Well, you know, dogs do that thing where they like scrape their butt on the floor sometimes, and then your kid crawls along and puts their mouth on the carpet. Boom. That's all it would take. So the moral of this story is that kids are gross and put some nasty stuff in their mouth, but also that that's important. That's how we get our microbiome. Now, the plants that we're talking about, these dicots, they're doing the same thing. Your mother doesn't directly pass her microbiome to you. However, when you're born, if you do a natural birth, moving through the vaginal canal, that's when you pick up your skin microbiome. And you get microbes from milk and from just putting junk in your mouth. So it's not exactly the same sort of deal as.
So what about if the mother eats something and then is breastfeeding and is able, can she pass it on that way to the child?
Yeah. Mm-hmm. But that would still be considered a horizontal transmission.
Horizontal, not vertical. Right.
Right. Mm-hmm. So I think I went on a tangent and maybe forgot the original premise of the question. Did that cover it?
Yeah, you covered it. She was asking if the thing. So let's see. If we have fungi living in us that usually plays nice, are the fungi in us considered endophytes?
Okay, yeah. So not endophyte, but endoanthro, I guess. I don't know the word for it. Yeah.
Yeah. Hopefully that answers your question. So.
Okay.
I have a question.
Mm-hmm.
So it talks about what is it, the Jansen-Connell hypothesis? Is that just, where does that come from? Is that just somebody who studied this in the past and come up with this hypothesis, or because it's just?
Yeah, it's named after the authors of a study. Let's see the citation here, 22, 44. So it's from Plant Ecology, paper in Science in 2012, conspecific negative density dependence and forest diversity. So that's a mouthful. The idea here is that if you drop all your seeds really close to you, you're not going to have as much fitness, because any diseases that you've picked up during your lifetime are also going to be falling with your leaves on your offspring. And so the idea here is that there's a selective advantage to broad dispersal. So you're kind of getting away from your parents' pathogens.
So they were able to kind of modify this hypothesis because they were able to find, even though they are maybe more subject to coming in contact with these pathogens, that the parents could potentially help them to become sort of resistant to whatever the pathogen is. Is that right?
Yeah, I think even more specific, yeah, you're coming in contact with the parent's pathogens, and since you're related to your parent, maybe you're likely to be susceptible to them also. But in addition to that, the parents are also passing on their beneficial microbes, right, horizontally, but they're still passing it along because you grew up in their litter. You're picking up the same beneficial microbes that helped out your parent. And so basically the results of this study seem to somewhat invalidate this Jansen-Connell hypothesis.
Okay, thank you.
At least make us rethink it.
So, Dr. Zhang, another question about the Jansen-Connell hypothesis was, when I was reading it, I guess maybe I misunderstood, but I took it more as instead of them getting planted next to their parent, where they would then have to worry about the pathogens, wouldn't it also have to do with having limited resources as well? Or was that just not part of the hypothesis? Like competing against their own family member, I guess?
Oh, so like intra-specific competition for... Yeah, and that gets to another hypothesis that we're still testing, which is Darwin's naturalization hypothesis, sometimes also known as phylogenetic overdispersion, where the idea is that you're not going to see a bunch of closely related groups inhabiting the same niche because they're going to have more competition with each other. And so natural selection ought to select for one of them to be better and then more distantly related things. So it's kind of like if you take any patch of environment and look at the phylogenetic relatedness of all the organisms that kind of fill that niche, it should be more distantly related than you would expect due to random chance. We don't know if that is the case in all groups.
So then, based off of that theory that you just mentioned, this is what had me thinking is when I was a little kid and you see the pine cones fall and like, where's the pine? It's like, what is the one plant that has like the seed that has like the spinner on it so it could fly farther from the tree? It had me thinking about those, you know?
Maples.
Yeah. And so that would then fit with, not with the Jansen-O'Connell hypothesis, with the, what do you say, the Darwin one?
Yeah, I think it might actually fit with the Jansen-O'Connell also.
Okay.
Because in maples, that little seed is called a samara, and it helps them get away from the parent in a strong breeze.
Okay.
The idea being that if a parent has been growing in one spot for a while, they've been accumulating pests and pathogens kind of in that area, and so the pathogen load for that should be somewhat higher than perhaps in a surrounding area where that species hasn't been growing. So the idea is if you can get away from your parent, maybe you can escape some of those accumulated competitors or pests. Here in this paper, they're kind of taking that and saying, but wait, right? If you do that, you're also maybe missing out on some of the beneficial microbes that your parents accumulated.
Okay, cool. Thanks, Dr. Zong.
See a couple hands.
Yeah, I've got a question about that as well. What? So it says that it kind of varies, like on the dispersal and stuff of it. Does that just depend on kind of like, for instance, like those maple leaves, that kind of the helicopter deal? Does it just depend on how they're dispersed, or is what kind of causes that variation with that hypothesis?
With the Jansen-Connell or the... Yeah. I think it's more of a... They went in and measured fitness of offspring that landed close to the parent tree versus those that landed further away, and found higher fitness in those that landed further away in their study. And so they hypothesized that the reason for that was that the parasite load was lower further away because they hadn't been building up. Obviously, some plants are better at long distance dispersal than others. I mean, look at coconuts. They float and can survive salt water for a year. That's good dispersal. And they get really far away. Other plants, maybe have big seeds that don't travel very far. There's the weird cannonball tree in the Pacific Islands. That's 2 feet away, and they drop a bunch and hope that some of them make it. So the dispersal mechanisms that different plants have evolved definitely play into how you would interpret this hypothesis. See,
Thank
Jeff.
you. Sorry, it wasn't a very good worded question.
No, that's all right.
Yeah, I had a question just because I guess I wondered, when I was reading about this paper, it reminded me a lot about the arbuscular mycorrhizal fungi and how sometimes that will like go into the nodes of the roots. And it's actually like inside of the plant itself. Isn't that kind of like what's happening in the leaves? Is this kind of like a further adaptation of that, or?
Definitely. I think it's safe to say that arbuscular mycorrhizal fungi are endophytes because they're growing inside of plant roots. They're just root associated, and here we're looking at the above ground part of the plant.
Cool. But this is also offering, I mean, I guess, does the roots also protect them from like pathogens and things like that as well, right?
Yeah. The reason we don't know as much about the above ground stuff is because for decades everyone's focused on the roots, kind of thinking that's where most of the pathogens are, and that's what we know about for, you know, all these plants have arbuscular mycorrhizal associations. A bunch of plants have ectomycorrhizal associations, which we'll talk about next week, which surround the roots. But what we're finding recently, you know, in the past 5-7 years, is that stuff associating with plants in their leaves might be just as important. All right, Leah.
So I had a question about Shannon diversity. It was one of the figures, and I wasn't 100% sure what that was.
Oh, yeah. Which figure was that?
It was in figure 2.
Figure 2, okay. I'll bring it up here, so we're going to look at it. Okay, so Shannon diversity is just one measurement of what we call alpha diversity. So if you imagine going into some plant and you sample its leaves, grow a bunch of fungi, and count the number of fungi that you find, in ecology we call that richness, just the number of different species. But that doesn't really take into account how they're distributed. So if you have, let's say you have 3 species, A, B, and C, and species A, you find 1 of them living in that leaf. Species B, you find 1, and species C, you find 98. Which one of those species is clearly dominating? Well, it's species C. There's 98 of them in that leaf and only 1 of the other 2 species. So the richness of that would be 3. And then you take another leaf and you find the same 3 species, but there's 33 of the first 2 and 34 of the third species. Which one of those leaves is more diverse, would you say? They both have a richness of 3.
The second one?
Yeah, it's more spread out. There's like more evenness. And so Shannon diversity is a way of incorporating the number of species that you have and how spread out they are amongst each other into a single number. And so a Shannon diversity, the higher you go, the more species and the more well dispersed amongst those species are the numbers, the counts of them. And so that's all that Shannon diversity is. It's just the richness plus the evenness.
Okay, that makes a lot more sense. Thank you.
Dr. Zahn, I have a question if no one else is asking anything.
Yeah.
Okay, so, and mine was more, okay, so in the paper, they talk about organizing, it says both the culture independent and the culture dependent data sets were organized in operational taxonomic units. And then they talk about OTLs and how they ran different statistics and whatever and what they found. What are OTL, like, is this something that they just used or is this something commonly used? I don't know what an operational taxonomic unit stands, because it sounds like there are several.
It's definitely jargon, and it's jargon that I forgot existed because I see it so often. So it's not something that they just use. It's definitely like an industry standard. What this arose from is the idea that, you know, with deep sequencing and like sequence barcode kind of stuff, we can go in and just amplify all the ITS1 region of fungi, and we get back a DNA read and a count of the number of times we saw it. And that DNA read, we didn't actually want to call it a species because we're not 100% sure that it is. It's really just a string of DNA that we found.
Right.
We can assign it to taxonomy if we're lucky. And so we can say it's likely this species or whatever. But since what we're actually measuring isn't a cell, it's not an organism we can see and hold, it's just a string of DNA, we call it an operational taxonomic unit. And so
Okay.
it's just a sneaky way of saying species without using the word so that we don't get into trouble.
And it's basically a way to look at bigger data sets or a lot of DNA basically at the same time when you're searching for just something specific, right?
Absolutely.
Okay. Awesome.
And so
okay.
we treat that string of DNA as if it is a discrete species.
Right.
But species has a meaning, and so we can't actually say species.
We can't say that. Okay, great. Thank you so much. Yeah, because it was not very clear to me. So
yeah, I'm sorry about that. I should have mentioned it first. If you do any microbiome work, you'll see the term OTU
so much
in every paper.
Mm-hmm. That makes sense. Now I can see why. Yeah, that's very helpful.
They may not, at this point, they may not even like tell you what it means. I've seen papers recently that don't even define OTU. They just assume that the readers all know what it means.
Kind of like DNA. We all assume we know what it is.
Like DNA. Yeah.
Yeah.
Exactly.
Got it. Okay. Thank you so much. I appreciate it.
Mm-hmm.
So with the Jansen-Connell effect or hypothesis, is there any evidence to say that one or the other is more beneficial when it comes to like dispersal? Or is it just kind of like, well, there's pros and cons, but it kind of evens
out? To my knowledge, no. To my knowledge, it has not been well tested. This paper is only a couple years old, really, and it's one of the first observations that kind of gives us a reasonable doubt about Jansen-Connell. I have not seen it. This could be my failing because I don't read many plant papers unless they're talking about the associated microbes, and so maybe I've just kind of missed it in the literature, but I definitely haven't seen it from a plant microbiome perspective being rigorously tested either way.
Okay, so this was kind of an important discovery, I guess, then.
Yeah.
And has there been any like pushback against it? I mean, I don't know. I mean, it makes sense to me, but I'm just curious about.
Not to my knowledge. Then again, it's maybe I'm just reading the wrong journals. In my extensive free time that I have to just peruse scientific literature, I haven't found time to like really dive into the plant ecology literature. Nor do I plan to necessarily. It's hard enough keeping up with my own field. I think if there has been criticism, it probably came from plant ecologists, but I have no awareness of it. I don't think they're making a really dramatic point about it, or any. It doesn't seem controversial to me. Like you said, it makes sense to you. It makes sense to me also. And their results definitely speak for themselves. Like the plants that grew up in cacao leaf litter did way better, right?
This might be a too general of a question, but when you say that there might be pushback specifically from people who are like plant ecologists, do you think there's any like bias that goes into like research like this, or people are like, no, but we found this, so we're sticking with this? And do you think there's like bias in the field at all?
Yes. Yeah, that definitely happens.
Cool.
During my grad school, for some stupid reason, I got really into the literature around like microbial dispersal, kind of like we talked about recently. Everything is everywhere and the environment selects, or there's like strong geographic patterns. And there were some serious arguments about that at meetings, and flame wars on Twitter, and people writing like competing perspective pieces in journals. And eventually I just kind of had to say, I actually don't care that much. I can't get worked up about this. But yeah, there was a lot of recalcitrance and people kind of seeing each other's point of view on that issue. It's the same for a lot of aspects of science. There's a theory, maybe you help develop it. It's kind of your baby. You don't want to see it be crushed. But at a certain point, evidence wins all arguments. Or should, yeah.
I have another question, I guess. Sorry, not related to what you were just saying, but related to the paper. So okay, so this paper kind of says that the Jensen-Connell hypothesis was wrong, right? Or at least not fully correct.
Yeah.
Has there been anything, are you aware if there is any other research more recent that proves if one or the other?
No. Yeah, that's what I was trying to convey with the previous question, is that this is kind of the last word that I know about it. And that's not super surprising for me. This is only from 2017. It seems like it's a wide open question and would be fairly easy to address with a more targeted study. Kind of do something like this with several different sorts of plants and kind of test the fitness.
It differs based on the species that's being tested, or.
I don't think it would be too difficult of a study, and it being 3 years old, I imagine that other people who read this, who were looking for a research question, probably noticed that and got to work on it. And so
we might have something soon.
Which would be the reason that I'd be reluctant to want to test it right now, is because as soon as you get into your project, somebody's going to publish your exact answer you were looking for, and damn it. So.
And it's the opposite of what you're hoping.
Yeah.
Potentially. Okay. Thank you.
Yeah, if you get a different answer than the people who just scooped your story, that's cool, because you can still get it published. But if you've got the same story in your data and they get it published first, you're kind of out of luck.
Yeah. I have a funny comment earlier when we were talking about the microbiome and getting it from dogs. I remember our conversation about the feces transplant, and I was thinking I would rather get it from dog than human. Just say.
Yep. I agree. This, I'll put a link. There's Popular Science article, I'll stick it in our chat here, that actually covers this paper, and that's how I became aware of it. This came out, when was it? Pretty soon after this, like 2018. Yeah, September 2018. to read that, I think this is from Science News. It requires you to like enter your email address. They don't send you anything, they don't charge you, but you do have to kind of like register as a Science News reader to get access to the article. But this covers several different studies that are all kind of looking at the same thing, which is trying to engineer plant microbes to accomplish some goal. So they talk about my Hawaiian study, they talk about this cacao study, and then an agriculture example from Indigo Ag, just to give you a feel for kind of what went into this. And you get to see some of the interview from the lead author of this study. She did this as a grad student, and it was just really great work. So I was a big fan of it. Any other thoughts, hopes, dreams, fears?
We're matching today, Dr. Zahn.
I'm sorry, what was that?
Said we're matching today. I'm wearing a backwards hat and a flannel.
Nice. I live in my bathrobe nowadays. I'm cool with it.
Nice.
Are there any other, this is kind of random, but are there any other websites or, similar things that you follow that you find interesting? I just signed up for the Science News, and I get articles from Science Direct. But I was just curious if there's other similar.
Oh, let me show you something pretty neat.
Can anyone else hear me?
Yeah, Dr. Zahn's screen froze.
Okay, so he's the one that got okay. I was like freaking out. I was like, oh no, right when I asked a question.
Yeah, I know. I'm like, where'd he go?
He's like, let me share something neat. Me disappearing.
John has left the chat.
It's not just my screen. He's actually, his screen froze.
Yeah, it's him.
Yeah, I can hear you guys. He's frozen.
Unfortunate.
His face is gone now.
I think he's restarting.
I'm back.
Oh, yay.
Sorry.
Yay!
Teams just like lost its mind. I tried to share my screen, so it may kill me again. Let's try this. Oh, okay, it's working. So if you're interested in following a particular topic, I recommend going to Google Scholar, and you can create a login in Google Scholar and essentially tell it stuff that you're interested in. So let's see here. Google Scholar alerts. There we go. So just search for Google Scholar alerts, and you can give it keywords. So I've got like phylosphere community, because I do plant microbiome stuff, microbial predation, plant microbiome, some authors that are pretty regularly turning out stuff that I find interesting. And so with these alerts, it will actually just send you a little email once a week saying, hey, here's some new exciting research with your keywords. And when you go to Google Scholar, it will start giving you recommended articles. And look at this. It's recommending plant diversity and litter accumulation mediate the loss of foliar and to fight fungal richness following nutrient addition. Neat. So that just came out. So seeing these recommendations is a good way of kind of keeping up with a topic that you're interested in. Now, to be honest, there's no way that I have time to read all of these, but if I see a title that I'm like, whoa, neat, I immediately just save it to Zotero. I'll read the abstract and just kind of keep the citation in my back pocket for when I am going to be writing a paper. So that can be pretty nice. You can follow authors on here also. Let's find. So let's see if Natalie Christian's done anything. University of Louisville. All right, so it looks like she is a professor now. You can sort her papers by year or by number of citations. And so you can kind of get a feel for things she's done since then. So here was that paper we just read. Oh, I just read this one. I didn't realize she was an author on it. Yeah. So following authors is, or at least just kind of looking them up on Google Scholar is another way to kind of see if more research has been done on that area, because they're going to be the ones to keep working on their system.
That is awesome. Thank you so much. I didn't know about it. Maybe other people did, but I have no idea.
Yeah, it's most people actually don't know that. I didn't learn until late in grad school that you could just get Scholar alerts, and
that's amazing.
They're really cool. It's kind of a pain in the butt, honestly. 9 p.m. rolls in on a Friday, and I'm ready to like watch The Office and fall asleep or whatever, and boom, there's an alert on my phone like, hey, there's 10 new articles. Oh, dang it.
No.
But it is nice. Okay, well, sounds like you are all out of questions. If so, have a good rest of your week. Nice weekend, and we'll see you Tuesday, and we'll talk about mycorrhizal fungi. Ectomycorrhizal, not the endo, so not the AMF, but the ectomycorrhizal. All right.
Until then.
Thanks, Dr. John.
All right, later.